Good morning from the West Coast, where it's quite early. I really appreciate the opportunity to present today. And just a quick disclosure, I am the co-founder of a company that is looking to commercialize certain aspects of the technologies I'll be talking about today. So what I'm going to be talking about today, as you may know, HIV infection generates a massive immune response, particularly a massive T cell response. In fact, it attacks the T cells itself, which is one of the reasons that it creates a persistent type of infection. But this massive expansion of T cells to HIV kills HIV-infected cells, but like I said, ultimately doesn't clear the virus from the body. And what my lab's interested in doing is understanding the mechanisms behind why this clearance is incomplete, and in ways in correcting these defects that exist. And then what I'm going to be talking about today involves the engineering of HIV-specific immune responses. And unlike current methodologies, which really right now focus on the engineering of T cell responses using peripheral blood or mature T cells, I'm going to be talking about the engineering of these cells through the blood-forming or hematopoietic stem cell. And the ability to look at this is really optimal in the humanized mouse models, because it is really a great model to look at human hematopoiesis. So engineered immunity, as you may know, is really a breakthrough approach for cancers and chronic viral infections. And it involves a genetic reprogramming of T cells using, and what I'm going to be talking about today is chimeric antigen receptors. It also uses molecularly cloned T cell receptors, but I'm going to be focusing on CARs today. And in this approach, T cells are redirected to specifically target and eliminate virus-infected or cancer cells through the use of these molecules. Current gene therapy implementations of this involves the reprogramming of peripheral blood T cells. Like I said, people take out peripheral blood T cells from an individual and then modify those cells and put them back in the individual. And this has really been effective only for liquid tumors like leukemias, but really it's been challenging in its success in solid tumors, and it really hasn't been successful in chronic viral infections such as HIV, which I'll be talking about today. And one of the reasons for this is these peripheral blood T cells are heavily processed, and when they're put back in the body, they're often abnormal and largely function only short term. And the reasons for this are many. There's a whole field investigating ways to correct these abnormalities and really enhance cell processing techniques to make this better. But what I'm going to be talking about today is one potential solution for this problem. So instead of using peripheral blood T cells, we're really focused on using hematopoietic stem cells. And these hematopoietic stem cells exist in the bone marrow of everyone and last a lifetime, and they continuously and naturally produce T cells in the body. So what the methodology I'm talking about is introducing chimeric antigen receptors into these minimally processed hematopoietic stem cells. So we're able to take these out of people and modify them using very, very minimal processing techniques, put them back into an individual, and then they engraft into the bone marrow to provide, much like they naturally do, to provide unlimited production of genetically reprogrammed T cells that would likely, or that hopefully will target virus-infected cells. And I'll show you the data that really suggests that this whole methodology works. This engraftment provides a lifelong supply of naturally developed and selected functional killer T cells targeting HIV. And we believe that this can overcome the defects caused by the cell processing and other limitations in the peripheral T cell approaches. So that we believe this is one solution to overcome limitations in that and really target chronic diseases, chronic viral infections and chronic malignancies, and really enable a long-term immunity to develop. So we've done a number of studies, and we have a number of previous publications detailing this use of this chimeric antigen receptor in humanized mice and in non-human primate systems. And the foundation of what I'm talking about really rested on a chimeric antigen receptor that was cloned initially identified and made about 20 years ago. And what this is an artificial T cell receptor that has an extracellular recognition domain. In this case, it's the CD4 molecule, which is the primary receptor to HIV infection. A transmembrane domain, usually by CD4 or CD8, and then that's fused to the intracellular signaling domain of the T cell receptor, the CD3 molecule. So when this is expressed on the surface of a T cell, it functions exactly like a T cell receptor. After it ligates its ligand, it sends a signal inside the cell for the cell to function. What I'm going to be talking about today is more or less the optimization of this whole approach. So we've done a number of studies with this full-length CD4 CAR. We've shown that you could put it into hematopoietic stem cells in mice and non-human primates, and these cells engraft in these animals basically for the lifetime of the animal. These cells are capable of forming multiple lineages that express the chimeric antigen receptor, including, importantly, T cells, natural killer cells, and macrophages, which are this receptor is known to function in. And so I'm going to talk about today the optimization of this approach. And we know that the full-length molecule has some limitations. One, it allows HIV infection of cells that may not normally be susceptible to HIV infection, particularly CD8 cells. So when it's expressed on the surface of this cell, which it should function on, it can allow HIV infection. And it also binds IL-16, which is a ligand to the D4 domain of this molecule, which is it's a chemokine and also a cell activation molecule that could non-specifically activate cells that express this. So we optimize this molecule by truncating it, eliminating the D3 and D4 domains, and basically trying to more optimize this molecule and its function by including costimulatory domains 41BB or CD28, much like they do in the cancer field. So we're trying to make this molecule more potent. And we've always used a lentiviral vector, knowing that CD4 can allow infection of these cells, a lentiviral vector that expresses two antiviral genes. These are shRNAs targeted to CCR5, which is a co-receptor molecule it uses to infect the cell, and the LTR and HIV. And we also have our various CAR genes also in this lentiviral base vector. So this really enables these whole studies to proceed. So we're really looking to optimize, and what I'll be discussing is the optimization of this chimeric antigen receptor. We're really trying to optimize the functionality of the cells and characterizing the functionality of these cells. When we made and expressed this D1, D2 truncated version on the surface of the cell, we found that just the expression of the molecule made the cells refractory to HIV infection. It didn't allow HIV infection through this CAR. This is a full-length CAR, and this, in fact, allows HIV infection by R5 tropic and X4 tropic, two different variants that use two different co-receptors of HIV. And, in fact, this D1, D2 CAR does not allow this. And when we made cells in humanized mice, and I'll discuss this and how we did this in just a second, and took them out and then stimulated them with, these are control cells. These are HIV envelope expressing cells, which bind the CD4 molecule on the CAR, or IL-16. When we stimulated the cells with envelope or with IL-16, we found the induction with the full-length CAR of interferon gamma and degranulation molecules CD107, actually very potently. And when we truncated in the truncated molecule, while it still responded to envelope, because it still needs to bind envelope and express interferon gamma and CD107, it didn't respond because of the lack of the domain to IL-16. So we believe this is a safety mechanism that is additional in this molecule. That enables the further development of it. So it doesn't non-specifically respond to pro-inflammatory cytokine IL-16, and it doesn't allow HIV infection. But we still have the antiviral genes in there because there are CD4 T cells that express these. We found that this truncated version has very similar in vitro function. So when we transfected CD8 positive T cells with these molecules and then stimulated them with, again, HIV envelope expressing cells, we found the stimulation of interferon gamma and TNF alpha very similar to the full-length molecule. And this molecule in a in vitro killing assay functions almost identically to the full-length version. So there's really no functional difference of this molecule compared to the full-length wild-type version of it. And we generate humanized mice. Many of you are familiar with the BLT model. So we take human fetal liver and thymus and plant it in the kidney capsule of an NSG mouse in this case. We take syngeneic CD34 positive cells, and in this case, modify it with our chimeric antigen receptors, and then allow the mice to develop 8 to 12 weeks. So these mice are basically pre-made with this chimeric antigen receptor, really for our proof of concept studies, to really to optimize this and really enable clinical implementation of this. We're doing additional studies to make these mice after infection, but we make these mice ahead of time in these studies, and we identify what is going on in these mice. So we like I said, we have a number of different publications showing that this is feasible with a full-length CAR. And in these mice, every one of these mice with a full-length CAR molecule, we tend to see a skewing towards B cell lineage development of the full-length of cells expressing the full-length CAR. And in the D1D2, we did not see this. We actually saw a slight skewing towards T cell development. And we think this is interesting and an improvement on what we're looking at doing. And we really want to make more T cells. I should add that this CAR molecule is not functionally expressed in B cells. It lacks the machinery to use this. So it's just an additional molecule on the surface of the cell. And the reason behind this skewing, we believe, is the interaction of the CD4 molecule with MHC2, a natural ligand of CD4 in the thymus of these animals. So basically, this full-length CAR enables MHC interaction to the point at which the cells are negatively selected. And what suggests this is when we looked at endogenous T cell receptor expression. So when these cells also express an endogenous T cell receptor in addition to the introduce chimeric antigen receptor. When we look at the cells that express the full-length CAR, we see a significant downregulation of the endogenous T cell receptor, where we don't see this with the D1D2 CAR. So this suggests that this chimeric antigen receptor is signaling and downregulating the endogenous TCR rearrangement. And backing this up here to the right, when we look at the cells that express the highest levels of this full-length CD4 CAR, we really don't find, noted by the red here, we really don't see anything in expression of the endogenous T cell receptor. So these cells expressing most of this chimeric antigen receptor are able to, in the process of developing, downregulate the endogenous T cell receptor and turn it off. And in fact, the only T cell receptor on the surface of these cells is the chimeric antigen receptor. And the D1D2 does not do this. It does not downregulate the endogenous T cell receptor. But we think biologically this isn't very significant, but it does suggest that the D1D2 doesn't preferentially select any type of T cell receptor molecule and doesn't skew the immune response in any way through this type of therapy treatment. So we actually think that the D1D2 CAR and its ability to allow endogenous T cell receptor to be expressed in the cells and improvements on function. And when we infect these animals expressing the D1D2 CAR, we see almost identical suppression of viral replication. Interestingly, we do not see inhibition of infection. And we believe this is due to the fact that HIV has just evolved to be so persistent and overwhelm the immune response so well that it is unable to, it outcompetes these basically naive cells at the point of infection to suppress it or to prevent it from being infected. But it does suppress HIV replication significantly over a log in most cases. And the function between the D1D2 and the CD4 CAR are very similar in this aspect. And we're looking at ways to improve this, and I'll go into that in just a second. But we really are looking at ways at basically immunizing an individual that contains this CAR in humanized mice, and really trying to shift these naive cells, which are slower to respond to initial HIV infection, to memory and effector cells, which respond much more rapidly. So we think an immunization strategy would likely have greater suppression or possible prevention of infection at this point. So when we look at we next look at the next generation of costimulatory molecules and hopes of enhancing the response of this chimeric antigen receptor. We looked at 4-1BB and CD28. That's also on this molecule, and made mice that contain different permutations of either the CD4 CAR or the D1D2 CAR with and without 4-1BB or CD28. And basically, when we put these cells into humanized mice, they all engraft. They're all found in the bone marrow throughout the lifespan of the animal. So this is basically sacrifice. So they exist. However, the animals that received the chimeric antigen receptor expressing the CD28 molecule did not develop into T cells, into B cells very efficiently, if at all. So for some reason, the presence of CD28 on this molecule does not allow the hematopoietic development of T cells and B cells, or really any other cell type. So some signaling aspect of that is a likely culprit, but we did find successful differentiation of each one of these CARs expressing the 4-1B molecule. And like I said, there's more of a skewing towards peripheral T cells with the D1D2 CAR expressing animals. And when we infected these animals with HIV, we found a more rapid suppression in the animals that received the chimeric antigen receptor with the 4-1BB. This is 2 weeks. They more rapidly suppressed than the ones that did not have the 4-1BB. And this is maintained 4 weeks here, where the animals without the 4-1BB molecule, these CARs basically caught up. So we see more rapid suppression of 4-1BB by 4-1BB CAR, and the difference between the CD4 CAR and the D1D2 are very similar. With this full-length CD4 CAR, interestingly, we see more of the development of effector memory cells during this infection. I believe this is 4 weeks after infection. We did not see as much effector memory development with the 4-1BB containing CARs, even the D1D2 CAR, but we did see greater development of central memory cells. And this is noted to occur with peripheral blood T cells that are modified with 4-1BB containing CARs that are targeted towards CD19 or CD20 treating leukemia. So we think that this is a possible mechanism of enabling these cells to form longer memory cells, which is critical in treating a chronic viral infection like HIV. And then this is summarized over here. So the 4-1BB or even the D1D2 CAR containing animals form more central memory cells and lesser effector type cells during this process, and they're still able to more rapidly suppress HIV infection. So again, the persistence of these central memory cells, we believe, is critical in targeting HIV infection. We treated these animals with antiretroviral therapy after about 5 weeks of infection, and we see suppression of these animals within 2 to 4 weeks following antiretroviral therapy, and this is a known way to treat HIV and has been largely successful in treating HIV-infected people. So this suppresses HIV. We found a more rapid suppression after the initiation of ART of animals that received the 4-1BB D1D2 CAR, and that's seen here in the pink. So in the magenta, I mean, basically this D1D2 CAR more rapidly suppressed after the initiation of antiretroviral therapy, and here at the tail end, when we released them from antiretroviral therapy, suppressed animals for slightly longer than the other animals. So we believe that this is the more potently functional molecule of what we've been looking at. And this is, in fact, our lead candidate up at this point. We looked at the presence of these CAR cells over time, before, during, and after the initiation of antiretroviral therapy. And these D1D2 CAR 4-1BBs actually persisted longer, even after the initiation of antiretroviral therapy, than the other molecules did. And we believe this is an improved functional aspect of this, even though You saw a slight decline during antiretroviral therapy. When you take away the antigen, the cells contract. The D1, D2 CAR 4-1BB didn't do it as greatly. And that's shown here. The 4-1BB CARs seem to persist longer. And the reason behind this is, like I said, even during the chronic infection, they form central memory cells, but during the antiretroviral therapy, we saw the greater the creation of central memory cells in the D1 and the 4-1BB expressing CARs, and that's shown here. So the greater percentage, the significantly greater percentage of central memory cells when you take away the antigen, we think is contributing to this persistence, and the more rapid suppression and the more rapid the more rapid or the suppression of cells once the antiretroviral therapy is released. So this formation of central memory cells we think is very important in the long-term maintenance of immunity and in this therapy to HIV infection. So in conclusion, our CD4 zeta chimeric antigen receptor hematopoietic stem cell therapy is safe and feasible for producing long-term functional antiviral immunity. We think this could be readily adapted, and we're currently looking at readily adapting it to malignancy treatments. Our improved molecule, the D1, D2 CAR, does not mediate HIV infection itself like its predecessor molecule, and it doesn't permit IL-16 stimulation, but has similar viral activities, antiviral activities as compared to the full-length CAR. So we believe this is a dramatic improvement and a safety improvement on this molecule. Because stimulatory molecule 4-1BB, but not CD28, allows successful hematopoietic differentiation of CAR cells. And this seems to also exist in other studies that we've done with other CARs. So CD28 molecules, for whatever reason, signaling during hematopoietic differentiation doesn't allow the selection of these cells or the differentiation of these cells into mature cells. 4-1BB enhances antiviral activities of the CD4 CARs, and the D1, D2 CAR T cells display better persistence during antiretroviral therapy. And like I said, the D1, D2 CAR is really our lead candidate going forward with IND development in other studies. So this next generation CAR with the 4-1BB molecule may improve safety and efficacy of this hematopoietic stem cell based CAR therapy. And so what this therapy really looks like when it's implemented in people is to isolate stem cells from patients. And this is done readily and easily by administering on day 1 or day 0 a compound that allows, and there's several that allows the migration of stem cells into the peripheral blood. So they're collected from the peripheral blood. That day they're introduced, CARs are introduced into these stem cells, and then the next day they're transplanted back into the patients where within a couple of months they form mature T cells that are capable of attacking HIV or what we're trying to target. And our goal here is really to reconstitute the cytotoxic T lymphocyte response in chronic viral infected patients and really try to cure HIV and try to really resolve a lot of the other chronic viral diseases and chronic diseases. So really this whole procedure could be done within about 3 days. I'd like to acknowledge my lab, particularly Valerie Razik, Angie Jen, Myra Carrillo, William Yu, and Heather Martin, and the other important members of my laboratory, the UCLA AIDS Institute, and everyone, particularly our funding sources. And I'd like to thank everyone for listening, and I'd like to thank Jax for having me. Thank you.